All right, I'm going to do a mini lecture on each of the 4 biomolecules. So it's carbohydrates, lipids, proteins, and nucleic acids. So we're starting with carbohydrates. When you think of a carbohydrate, you are generally thinking of a sugar, and you might be thinking, well, what about bread? And bread is certainly carbohydrates as well. It's just slightly more complicated than that. In bread, it usually is residing in the form of things like starches, which are just larger polymers of some of these smaller carbohydrate subunits. So a thing called a monosaccharide is where we're starting with carbohydrates, and that means one sugar. Mono meaning one, saccharide meaning sugar, one sugar. It's just one unit. One unit of something with the molecular formula CH2O. And the CH2O is going to come in different multiples and determine actually what kind of carb we're looking at. So I have glucose and fructose listed here. You would consider these monosaccharides or single sugar units. And you see that they have the same formula. Well, as we're going to see in a moment, these two are actually what are referred to as isomers of one another. They have the same number of carbons, same number of hydrogens, same number of oxygens, but they're actually shaped slightly differently. And honey has both of these glucose and fructose monosaccharides within it. When you take a carbohydrate and you put it into an aqueous solution, aqueous just kind of, you can think of it as water containing. When you put it into something that has water in it, they form rings. As we're going to see in a moment, they spend a lot of their time in these linear formats, but when you put them into an aqueous solution, they do form these rings. Another important detail here is that carbohydrates are the main fuel that your cells are using to do work. So as we're going to find out later, what your cells do is they break down these carbohydrates and they use them to create a chemical called ATP. And your cells are going to use ATP to fuel all of the cell stuff that it wants to do. And you can chain these things together and form what's called a disaccharide. Di meaning two, saccharide meaning sugar, or two sugar units together with one another. So, like I was talking about a moment ago, you have these things called isomers. Here you have glyceraldehyde, and over here you have dihydroxyacetone. And they have the same chemical formula. They have 3 carbons, both have 3 carbons. They have 2 OHs, each has 2 OHs. They have 1, 2, 3, 4 hydrogens, and they have an oxygen. So these are the same chemical formula, but you can see that they are indeed different shapes. They are sort of different variations on the same theme. You could think of the same chemical formula, different shapes. This is important for carbohydrates because over here on the left you have glucose, and then over here on the right you have fructose, which is the same exact chemical formula. Chemical formula is exactly the same, but they look different. So isomer just means same chemical formula, different structure. So this carbon has the carbonyl carbon up here on top, the double bonded oxygen, I should say, up here on top. The fructose has it kind of in the middle. Same chemical formula, different thing. So, like I was saying earlier, these tend to be drawn or depicted linearly. They don't tend to be depicted linearly, but they are often depicted linearly, but they can form these ring structures. And they'll do this, they'll form these ring structures usually when they are in the presence of water or in the presence of something polar. So I told you guys we weren't going to do organic chemistry. I just wanted to show you this. This is an abbreviated version of the ring structure. So everywhere that there is a point on this ring, everywhere that there is a point, there is a carbon. So there's a carbon here, there's a carbon here, there's a carbon here, there's a carbon here, and there's a carbon here. There's no carbon here because that's where the oxygen is. Everywhere there is a point, there is a carbon, and everywhere that there is a carbon, it is also suggesting that it's bonds are all filled up by hydrogen. This is actually not my favorite slide. I got this from the book just because there are OHs coming off here, which really ought to be drawn. There's like an extra carbon coming off here, which really ought to be drawn. But the main point I want to get across is everywhere that there is a point or two segments that are kind of intersecting with one another, there is a carbon and all of its associated hydrogen. So here are just a couple of very common carbohydrates. That's glucose and fructose drawn out every molecule. You'll notice that this is sort of a five-pointed shape here, and this is more of a six-pointed shape here. And it also is showing you the space filling model that I was talking about earlier that kind of shows you the size that this thing, or the space that this thing is actually occupying. So, like I was saying earlier, you can indeed take two units, two monomers, two monosaccharides, and you can put them together to form a disaccharide. So this is a disaccharide. There are two monosaccharides here. And like we talked about in the other lecture, what you're going to do is dehydrate these two. You're going to remove the water and cause these two to hook up into this disaccharide. You also have polysaccharides, which are these complex sugars that have many sugar units. An example of this is starch, which is a whole bunch of glucose molecules all chained together. And starch is a common energy storage thing used by a lot of plants. So lots of potatoes, lots of different plants use starch. You also have glycogen, which is glucose molecules chained together, but in a slightly different way than starch. And this is what animals are using for energy storage a lot of the time. And by the way, these organisms are not just using carbs for energy storage. They're using lipids and other things as well. And you also have cellulose, which is, again, just glucose molecules chained together in a different way than these first two. And this is a structural molecule that is used in plant cell walls, like we saw in lab that first day. So here is starch, and it's just showing you this is a bunch of glucose molecules kind of all chained together with one another, and you will find this inside plant cells. Here is glycogen, which is, again, animal cells, and you can see it's just a bunch of glucose. These green trapezoid things, they're not trapezoids, pentose things are it's not even pentose, hexose. These green hexose shapes, these green shapes are those glucose, and it's just showing that they're all chained together, and this is what is used in animal cells to store energy. And then finally, you have these cellulose fibers. Again, you're seeing the same pattern here, these same glucose molecules all chained together with one another to form the cell walls of plants. And these cellulose things are very often what you see in dietary fiber, because we can't really digest them, so they help us to sort of clean out our digestive system. So next we will move on to lipids.